Hello, my name's Dr. Selina Cox. I'm a postdoctoral research associate at King's College London, and currently I'm working on dietary interventions for managing Crohn's disease. I'm going to be talking to you today about the various dietary approaches that we have for the management of IBD, but I'll be particularly focusing on what the future might hold for the dietary treatment of IBD. I felt that it was important to first mention any sort of associations that we have between dietary factors and the development of IBD. Patients often have questions around whether something they ate in the lead up to their diagnosis caused them to develop IBD, or indeed whether something they've been eating throughout their life has caused them to develop it. And we do have some prospective studies that have investigated have recruited groups of individuals from population, followed them up for a diagnosis of IBD, and then looked at their dietary intake preceding their diagnosis. Some of these studies have shown some associations, but on the whole, it's very difficult to establish any cause and effect. Well, it's impossible to establish cause and effect relationship with these studies. So although some have shown that, for example, a high intake of fruit and fiber from fruit and veg may reduce risk of IBD, it's impossible to say whether that's related to their subsequent diagnosis or not. And then it's also important that we look at associations between dietary factors and IBD course. So are there any foods that people eat that may make them more likely to have a relapse? Again, this is something that I find that lots of patients really want to know about, because if there's something they can do or something they can avoid in their diet or eat in their diet that may prevent them relapsing, then that's often a really attractive option. Unfortunately, though, this is much the same situation as the previous slide, where there are some prospective studies that show that it may be prudent to increase fruit and vegetable intake for Crohn's disease, and to increase oily fish, or to increase omega-3 intakes for particularly in UC, and then for both Crohn's and UC, reducing saturated fat and trans fats may be useful. However, again, we don't have the ability to establish a direct cause and effect relationship here. But these recommendations are not too wild. They're not anything crazy. So I think increasing fruits and vegetables, increasing omega-3 intakes, and reducing saturated fat intakes is not a bad thing, particularly when patients are in remission. So first, I just wanted to touch on dietary treatment that we do currently use in IBD. This won't be news to most people listening, but the main treatment that we have currently is exclusive enteral nutrition. And as many will already know, this has been shown to be as effective as steroids in inducing remission, and it's the first line treatment for active Crohn's in children. But the problem with exclusive enteral nutrition, as anyone who's worked clinically with it will know, it's very difficult for patients to tolerate, and it's very difficult to continue to comply with over an 8-week course, which is what's recommended. There are several outstanding questions with regards to exclusive enteral nutrition, and I think one of the main ones is how it actually works. We're still not sure how it may put somebody into remission, and furthermore, some studies have shown it to be associated with mucosal healing, and again, the reasons for that are unclear. There's questions around whether it's something to do with the microbial changes that it causes, but these have been shown to shift more towards a dysbiosis, which is something that is characteristic of Crohn's and UC. So it seems counterintuitive that a dysbiosis would be associated with going into remission. So that's something that requires further research. Is there anything within exclusive enteral nutrition that might be inducing remission? I mean, that seems unlikely because different formulations of EEN have been shown to have no difference in terms of to all induce remission to the same extent. Or is it something to do with dietary components that are avoided when patients are taking EEN? Another question that we have is whether we could develop a solid diet, which would be easier to follow, but that mimics EEN. So a diet that that sort of matches EEN, and whether that could even be potentially useful in UC as well as Crohn's. And finally, a major question that I know lots of people clinically might have is what's the best food reintroduction diet? Some of the ones that are used currently are low flex, which is, excuse me, low fat, low fiber diet. There are reintroduction diets that are over 3 days. And then there are some that are a longer reintroduction, reintroducing, say, 1 or 2 foods each week, for example. So another dietary treatment that we have for use in IBD now is the low FODMAP diet. So this is particularly useful in patients with inactive Crohn's and UC, but people who have IBS-like symptoms alongside their IBD. And this is common in patients in remission and can affect up to 33% of patients, and can have a really significant impact on quality of life. And some patients report that these symptoms can be as severe as the symptoms that they experience when they're having a flare of their IBD. And so it can be difficult for them to distinguish between the two. We're really interested to see whether a low FODMAP diet, which is so effective in IBS, whether it could be as effective in patients showing IBS-like symptoms, but with a diagnosis of IBD. We first did a re-challenge trial, which involved recruiting patients with IBS-like symptoms in IBD, or functional symptoms, you might call them, and asked them to follow a low FODMAP diet until we had adequate relief of their symptoms. And then we gave them some pure FODMAP drinks. So there was a fructan challenge, a galacto-oligosaccharide challenge, and a sorbitol challenge. And these are all different types of FODMAP. And what we showed was that particularly the fructan challenge increased severity of pain, bloating, and flatulence. And that's once those symptoms had been managed on a low FODMAP diet. So effectively what this showed was that fructans were able to induce symptoms in patients with the functional symptoms in IBD. Following on from this, it was important then to establish whether a low FODMAP diet could be effective for managing these functional or IBS-like symptoms in IBD. And in this trial, we randomized patients with IBD to either a low FODMAP diet or a sham diet, which was designed to allow patients to follow dietary restrictions, but we knew from pilot studies that those restrictions didn't affect their FODMAP intakes or any other nutrient intakes. What we showed here was that the low FODMAP diet did indeed reduce global gut symptoms, but it also reduced members of the gut bacteria that are typically known to be anti-inflammatory. So those were mainly bifidobacteria and Faecalibacterium prausnitzii, and the latter of those is heavily associated with remission in IBD. So higher levels of F. prausnitzii may predict IBD remission. So it's important that we bear in mind that if somebody's following a low FODMAP diet, they do need to follow the reintroduction process after their restriction phase in order to allow gut bacteria to recover. And we don't know what the long-term effects on the gut bacteria would be of following a strict low FODMAP diet. I just wanted to mention the specific carbohydrate diet, which is something that some patients follow, and I've come across a few patients that do it and feel that it helps their symptoms. This was originally pioneered for celiac disease, but then was popularized for the use in IBD by Elaine Gottschall, whose daughter had UC and who claimed that it had essentially cured her of all UC symptoms and inflammation. The diet, the basis of the diet is a restriction of sugar, effectively, so disaccharides and all complex carbohydrates, so grains, legumes, potatoes, and the only sugars allowed are monosaccharides, so glucose and fructose. This is quite a restrictive diet, allowing mainly fruits and vegetables, some legumes, meat and fish, and eggs, and certain dairy products. The proposed mechanism, although not sort of established, not formally established, is through correcting a dysbiosis. So it's proposed by advocates that the diet may change the gut bacteria and correct the dysbiosis associated with the IBD. A number of mostly prospective and retrospective studies have investigated the specific carbohydrate diet. And generally, these do show improvements in IBD symptoms, but they're not randomized controlled trials. Some of them are very small studies. Most of them don't assess nutrient intakes. But one randomized trial has been very, very recently published, and this compared a 12-week specific carbohydrate diet to a Mediterranean diet in almost 200 people with Crohn's disease. So this was actually quite a large study. Patients had mild to moderate inflammation. This was actually essentially a comparative effectiveness trial, assessing whether one of the diets is better than the other, but there wasn't a control which would be expected not to affect symptoms at all. So it's not a randomized control trial as such. But what they found was that the specific carbohydrate diet and the Mediterranean diet were equally effective in inducing remission. So that does suggest that around 50% of patients went into remission with the specific carbohydrate diet. So that's the first randomized trial evidence of potential effectiveness, but further studies are needed comparing this to a true control, which would be a treatment that wouldn't be expected to improve symptoms. Sorry, there we go. Another diet that's a potential future approach for IBD is the Crohn's disease exclusion diet. So as we know, exclusive enteral nutrition is currently, I suppose, the most effective treatment for active Crohn's, a dietary treatment. And so these Ari Levine's group in Israel have attempted to establish a solid diet that could be used alongside partial enteral nutrition to have potentially the same effects on inducing remission. So the diet involves 50% partial enteral nutrition alongside diet excluding gluten, dairy, animal fats, processed meats, emulsifiers, and canned goods. They've assessed this diet in 74 children with mild to moderately active Crohn's. And in this study, patients were randomized to one of two arms. In the first arm, they followed a 6-week 50% enteral nutrition, 50% Crohn's disease exclusion diet, and then went on to 6 weeks Crohn's disease exclusion diet, reducing to 25% enteral nutrition, so increasing the amount of solid food for the second half of the study. And the other arm, it was effectively comparing to the standard treatment, which is exclusive enteral nutrition. So 6 weeks of exclusive enteral nutrition, followed by 6 weeks free diet, alongside 25% enteral nutrition. So I suppose essentially weaning off the exclusive enteral nutrition in that group. What they showed here was that, importantly, 97.5% of patients tolerated the Crohn's disease exclusion diet alongside the partial enteral nutrition, and that was compared to 73% in the exclusive enteral nutrition group. So it was significantly better tolerated. So having some solid food or half and half solid food and exclusive enteral nutrition was better tolerated, which I suppose isn't surprising. At week 12, 75.6% of patients were in remission on the Crohn's disease exclusion diet and partial enteral nutrition. And that was compared to 45.1% in the exclusive enteral nutrition group. And that may feasibly have been related to adherence, so better adherence in the group that were allowed to eat food. So this is really promising and shows that there may be an alternative to exclusive enteral nutrition that may not be as difficult for patients to follow. So watch this space. This diagram I just felt was a really lovely graphical abstract published with this paper, which basically illustrates the potential mechanisms of how the Crohn's disease exclusion diet might be working. And they've proposed that that's mostly likely due to changes in the gut bacteria. However, what is worth bearing in mind that this study was in children only, so there's no evidence so far of the Crohn's disease exclusion diet in adults, and that's, I would imagine, possibly the next stage for this work. Another diet that's been proposed is a entirely solid diet mimicking exclusive enteral nutrition. So this research group in Glasgow, and they've basically recreated exclusive enteral nutrition with whole foods and matched it for macronutrients, vitamins, minerals, and fiber. They've investigated this in a number of ways, and this was published in 2019. First, they investigated the diet, it's called the CD treat diet, compared to exclusive enteral nutrition compared to normal standard chow in rats. They then did a crossover study in 25 healthy people who followed a Crohn's disease, CD treat diet versus exclusive enteral nutrition. They did 7 days of each with a 14-day washout, and all their food was provided. And this was to investigate mostly whether the CD treat was more acceptable and tolerable, better tolerated. But also they did look at the microbiota alterations on the two diets. And what they showed was that, again, probably unsurprisingly, the CD treat diet, being based on whole foods, was better tolerated and more acceptable to the healthy controls. And actually, interestingly, it led to similar microbiome alterations compared to EEN. So this suggests that maybe they might be working in a similar way, which is promising given that they've aimed to recreate EEN. And then finally, they did an open label trial in 5 children with active Crohn's disease, where they followed the CD treat, the solid diet, for 8 weeks, where again, all their food was provided. And 2 of 5 children were in clinical remission at 8 weeks, while 3 out of 5 had a clinical response. So again, that's really promising. That was in patients with Crohn's, was only in children, and it was only in 5 people, and was an open label study. So although that's really promising preliminary data, most certainly the next step would be to do a randomized control trial comparing CD treat to EEN in both children and adults with Crohn's. And again, there's another graphical abstract that shows what happened in the different groups in this study. So in the rats, in the healthy volunteers, and in patients with Crohn's. But something to bear in mind here is that they provided all foods to participants, which is great in terms of encouraging compliance to the intervention. But it's not representative of clinical practice. Obviously, we wouldn't send patients away with all of their food for 8 weeks. We just wouldn't have the resources to do that. And so this effectiveness may not represent how effective the diet might be clinically. Furthermore, as I said, it is only in a small number of patients, children with Crohn's disease. There are also a couple of so-called anti-inflammatory diets that have been proposed for IBD, and these are generally based on increasing omega-3 fatty acids. There's also an increase in fruits and vegetables, and they often also advocate having fermented foods such as kefir, miso, and live yogurt. And so for example, the IBD anti-inflammatory protocol diet has been studied only in a retrospective study in 11 patients, so a small retrospective study. And to be honest, the rationale for the certain aspects of the diet is not particularly strong. So why are they excluding the things that they are? What's the basis for that? It's not clear. And then another anti-inflammatory protocol diet has been proposed as well. Again, only assessed in a pilot study in only 15 patients with active IBD. Remission was achieved in 6 out of 9 patients, but again, it's not a randomized control trial. And again, the rationale for excluding certain things, so for example, nightshade vegetables, was unclear. So I just wanted to talk about food additives for the final part of my talk. And one thing that all of the diets that I've spoken about so far have in common is that they all lead to a reduction in ultra-processed food intake and therefore food additive intake. And a couple of recent abstracts have been published which show that ultra-processed food intakes may be associated with IBD risk. So a couple of prospective cohort studies have shown that increasing intakes, increasing servings of ultra-processed food intakes per day are associated with an increased risk of developing IBD. So that's really interesting, and the sort of epidemiological data, but doesn't necessarily establish a direct relationship. So I wanted to talk mainly about emulsifiers here, and that's because these are additives that have been, I guess, most consistently studied in relation to Crohn's disease, mostly in recent years. Now, emulsifiers are substances that are added to foods or that occur naturally that allow an oil and a water to mix together. So normally they would repel each other and separate. As you can see on the left, that's a mayonnaise that's separate, whose oil and sort of aqueous phases have separated. And then with the addition of an emulsifier, which has a hydrophobic tail and a hydrophilic head, it's allowed to form a nice smooth mixture. So basically a palatable mayonnaise. These additives are found in a number of different types of foods in the UK, particularly in products such as ice cream, frozen and chilled puddings, breads and baked products, confectionery and ready meals, but also in other places that you might not expect. And there are over 60 emulsifiers in the UK food supply, so quite a lot of different types. Food additive emulsifier intake has been associated on a population level with Crohn's disease incidence, as you can see here in different countries in the world. And then the diagram on the right is in Japan, actually. So again, a recent abstract has shown again that early life, this was a case control study, and it showed that when comparing patients with Crohn's disease to their first degree relatives, as well as to their household members and to unrelated completely healthy controls, people with Crohn's disease had greater early life fast food consumption and processed meat consumption than their household members, non-family household members, and compared to healthy controls. And when looking at more recent intakes, intake in the last 12 months for patients with Crohn's disease showed greater emulsifier intake, sweetener intake, and total food additive intake than their first degree relatives. Now, again, whether this is some sort of reverse causation showing that people eat more of these with a diagnosis of IBD, or whether it causes or is related to the development of IBD is unclear. But this is a really interesting association. Now, I just wanted to look at the proposed effects of emulsifiers on the gut microbiome and on inflammation, and why these are being associated with IBD and studied in IBD. So Benoit Chassaing and his lab have worked extensively on emulsifiers in animal models and in vitro models. And what they've shown is that in particular, they've studied a couple of emulsifiers, polysorbate 80 and carboxymethyl cellulose. And as you can see here, this is an in vitro model, which is a model mimicking the gut, mimicking the human gut. And what they did here was that they spiked it, they gave basically inoculated the model with human microbiota, human stool, basically fecal samples, and then spiked those samples with polysorbate 80 and carboxymethyl cellulose. And what they showed was that the polysorbate 80 in particular caused the gut microbiota composition to shift away from the composition when given carboxymethyl cellulose and compared to when given water as a control. So basically, it seems to significantly impact on the microbiota that are in the model. Furthermore, both polysorbate 80 and CMC impacted microbiome transcription of pro-inflammatory factors. So what this suggests is that the emulsifier treatments were potentially increasing the pro-inflammatory potential of the gut bacteria. They then looked at the effects in mice, and what they showed here was that both emulsifiers reduced the diversity of gut bacteria in both wild type mice and IL-10 lacking mice. And the gut microbial diversity is something that is associated with active IBD. So this is interesting that the treatment with these emulsifiers in mice is associated with reducing diversity, which we would typically associate with active disease. And then also in terms of the gut barrier, they saw that CMC and P80 both increased the number of bacteria that were able to encroach close to the epithelial layer. So in these diagrams, you can see that in blue, you've got the epithelium, and in red, you've got the bacteria. And with the water treatment on the two left panels, you can see that the bacteria stay well separated from the epithelium. But then when you look at the CMC and the P80 panels, you can see that the bacteria are getting closer to the epithelium. So treatment with those emulsifiers in the mice appeared to impair the barrier in some way. Furthermore, it also increased both the presence, the incidence and severity of colitis in these mice as well. So why are emulsifiers having these effects? What are the proposed mechanisms? This is really well summarized in this diagram, which is in a review that was published recently by our research group at King's. And what the diagram shows is that the emulsifiers are present in the diet. These may increase the abundance of potentially pro-inflammatory bacteria through shifting microbiota composition, but also the expression of pro-inflammatory components of the bacteria, such as lipopolysaccharide A. And then furthermore, the emulsifiers may also damage the gut barrier and increase permeability, therefore meaning that the bacteria may be reaching the epithelial layer and therefore triggering inflammatory pathways, which might then lead to colitis. So that's the current working mechanism, and further research is needed to investigate these mechanisms, but that's currently what is being proposed. So have emulsifiers been tested in human IBD? So far, research in this area is very limited, and only one randomized control trial has been conducted. And in this trial, 12 patients with ulcerative colitis only were randomized to follow either a carrageenan-free diet or a carrageenan-containing diet. And the way they did this trial was to ask patients to eliminate sources of carrageenan in their diet. So everybody on the trial eliminated carrageenan-containing products. They were just given a list of foods to avoid. And then they were given either they were given either carrageenan supplements to bring their carrageenan levels back up again so that they were the carrageenan containing group, or they were given dextrose tablets. So they were effectively the no carrageenan group or the low carrageenan group. So they were essentially the treatment group in this case. What they showed here was that in the carrageenan group, there was greater rates of relapse of UC. So 3 out of 5 patients relapsed, and that was compared to 0 patients in the dextrose group or placebo group. And then there was also a worsening of health-related quality of life in the carrageenan group. But this was a very small trial of only 12 patients. It was analysed per protocol, meaning that patients who didn't complete the trial were not analysed. And it was only carrageenan restricted. So there are more than, as I mentioned, more than 60 emulsifiers in the UK food supply. Carrageenan is only one of those. And we don't know what the effects would be of restricting all of them. And it only included UC and not Crohn's. There are challenges that come with investigating a low emulsified diet in IBD. This is a really new, novel diet, which is difficult because there are, again, like I say, it's probably my third time saying now, there are more than 60 emulsifiers in the UK food supply. So it's very difficult to restrict that number of emulsifiers. But also identifying them is very difficult because there's very little consistency in the labeling of them on foods. They can be called different things. They can be listed with different permutations of the name. So for example, carboxymethyl cellulose can be written in many different ways. Sometimes the E number is given rather than the name. So all of this makes it extremely difficult for patients to follow a restriction of emulsifiers. Furthermore, we don't know how much people are eating of these emulsifiers or the actual quantity, because manufacturers don't have to put the quantity in the ingredients list. They only have to state that the emulsifier is present in the food. So our research group, in our research group, we developed a smartphone app that allows patients to to follow the low emulsified diet by scanning foods, and then the app tells them whether or not they can have that food. We tested this in 20 patients with inactive Crohn's disease to establish whether it's feasible to follow. They did it for 14 days and also had dietitian counselling alongside the app. And what we showed was that there was a 94.6% decrease in emulsifier containing foods in those patients for those 2 weeks. So a significant proportion of the emulsifiers were reduced. So it did show that using the app and with a dietitian, it seemed that patients were able to follow the diet. But what are our unanswered questions then with emulsifiers in IBD? So far we've answered the question that emulsifiers do seem to cause some inflammation and change the gut microbiota in animals and in vitro models. They do seem to impair the gut barrier, at least in animals. And it does seem that a low emulsified diet would be possible and feasible in Crohn's disease. However, we don't know if all 60 or so emulsifiers cause inflammation, whether they all change the gut microbiota and whether they do so in a similar way to each other. Again, we don't know whether these emulsifiers have the same effects in humans as they do in the animal models that we've seen so far. And we don't know what the mechanisms of effect are in humans. As I mentioned, it's so difficult to estimate intakes of emulsifiers because we don't know how much of them our manufacturers are using. So we need to investigate what are actually UK intakes of these emulsifiers. And furthermore, and importantly, whether a low emulsifier diet can reduce IBD activity. And then also, could it maintain remission? Could it be something that patients could follow in the long term to keep them in IBD remission? And finally, what about other food additives? Emulsifiers are only one class of additives, and there are many others such as sweeteners and preservatives that may also have an effect. However, we don't know about any potential mechanisms of these, and this is something that most certainly would need to be looked at in the future. So just bringing it back to recommendations, these are guidelines that are produced by the International Organization for the Study of IBD, and they suggest that it may be prudent to limit the intake of polysorbate 80 and CMC, which are the two emulsifiers that have been investigated most in animal models of IBD. As well as carrageenan, which, as I said, has been investigated in one human study in UC, but has also been looked at in a number of animal studies, which I wasn't able to mention here in the interest of time. And then they also mentioned titanium dioxide and other nanoparticles. But they are suggesting that patients do reduce certain emulsifiers. So that does suggest that we are moving forward in this area. And in terms of other emulsifiers, research will show whether these have the same effects on the gut microbiota and inflammation. And we are, in our research group at King's, currently doing a randomized control trial comparing a low emulsified diet, reducing all 60 emulsifiers compared to an emulsifier containing diet. So watch this space with regards to the results of that study. So in summary, what we've talked about is that in remission, patients may be advised to increase fruits and vegetables intakes, particularly in Crohn's disease, and increase dietary sources of omega-3, particularly in UC. And reducing saturated and trans fatty acids intakes in both Crohn's and UC. And as I said, these are reasonable suggestions and in line with the general population. But we would need to take into consideration any patients that have strictures and anyone who has functional symptoms who might potentially benefit from a low FODMAP diet. So as I said, a randomized control trial has shown that low FODMAP diet may reduce functional symptoms in people with IBD in remission, but it's really important that we encourage food reintroduction and not just following a low FODMAP diet in the long term, because we don't know what effect that would have on the gut microbiota. In patients with active disease, we know that EEN is effective for active Crohn's, but there are difficulties around compliance and tolerance. So what might the future hold? Well, we know that there are studies now published that have replaced some of exclusive enteral nutrition with an exclusion diet, or indeed replacing the whole, all of the liquid with a whole diet that mimics it. And there are also some studies, very limited studies, assessing anti-inflammatory diets, but a lot more research is needed in that area. And we're unsure of the role of emulsifiers in IBD, and that's something that is undergoing ongoing research. And there may be some in the future, some investigation of other additives as well. So that's me. Thank you very much for listening. And I'd be happy to take any questions over email should anybody have any.